Assalamualaikum and good day, everyone. So now we are going to learn about skeletal muscle relaxants or neuromuscular blocking drugs. The learning objective of this topic are to describe synaptic transmission at the neuromuscular junction and skeletal muscle contraction, and to list the clinical uses of skeletal muscle relaxants, to describe how neuromuscular blocking drugs works, to list the adverse effects which associated with NMB drugs, and also to compare and contrast depolarizing and non-depolarizing neuromuscular blockers. So first, we are going to look at to the first learning objective, which is to describe synaptic transmission at the neuromuscular junction and skeletal muscle contraction. We know that peripheral nervous system is further divided into somatic and autonomic nervous systems. So in this lecture, we are going to focus on the somatic nervous system. So if you still remember, somatic nervous system consists of motor division or afferent neurons and sensory division, or sometimes we call it as afferent neurons. Somatic nervous system is responsible in controlling skeletal muscle tissue, or in other words, it is responsible for voluntary movement. Muscles are controlled by the nervous system via innervation of motor neurons. In order for skeletal muscle to contract, it must first be stimulated by a motor neuron. And the gap, or sometimes we call it a space between these motor neurons and the skeletal muscle, we call it as a neuromuscular junction. So when an impulse travels between this space, muscle contraction can happen. So motor neurons instruct skeletal muscle to perform series of action that lead to sliding filaments and muscle contraction. Neuromuscular junction consists of a neuron and also a skeletal muscle cells. And the motor neurons who originate from the spinal cord innervate the skeletal muscle fibers. And this innervation happens by very fine processes of the axon. And the synapses are present along this process and are also known as motor end plate because of its very specific structure. NMJ synapse has three characteristic features. There are two membranes called the pre and also post synaptic membranes, and the uh, distinct space between these two membranes we call it as a synaptic cleft. High density of small spherical vesicles are present here, which contain neurotransmitter substances, and a thickened postsynaptic membrane is present, which has a very high density of receptors that are very that are responsible for the binding of the chemical substances which transmit the signal from the presynaptic neuron. This topography or this figure shows actually the neurotransmitters released at the neuromuscular junction. So how motor neurons excite skeletal muscle fibers? Motor neuron depolarization causes action potential to travel down the nerve fiber to the neuromuscular junction. And the depolarization of the axon terminal causes an influx of calcium, and which trigger the fusion of the synaptic vesicles and the release of neurotransmitters such as acetylcholine. And acetylcholine will diffuses across the synaptic cleft and bind to the postsynaptic of acetylcholine receptor, for example, which located on the muscle fiber at the motor end plate. This process opens an ion channel through which sodium ion passes to the muscle cells. At that time, the potassium ion diffuse out of the muscle cells. However, the amount of sodium ions entering is higher than the number of potassium ions leaving. Once the sodium ion reach at the muscle cell, it depolarizes. So action potential being triggered. The binding of acetylcholine to acetylcholine receptor opens the channel, causing an influx of sodium. and depolarization of the sarcolemma that travel down the T tubules and ultimately causes the release of calcium ion from the sarcoplasmic reticulum. So contraction can occur. Acetylcholine does not remain in the synaptic cleft of the neuromuscular junction forever. One enzyme called as acetylcholinesterase, or in this figure in a blue color, is responsible in acting as an catalyst in breaking down the acetylcholine present in the synaptic cleft, and it can give rise to the acetate and choline, which are then transported back to the synaptic cleft, where they are resynthesized, and this is what we call as an active reuptake. This video is about the sliding filament mechanism, which involve in the skeletal muscle contraction.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
This figure shows the mechanism of muscle contraction. So now we move to the second learning objective, which is about clinical uses of skeletal muscle relaxants. Drugs that affect skeletal muscle function. Basically, there are two therapeutic groups, those that are used to reduce spasticity, or in other words, as stiffness, and also spasms, which involve the centrally acting muscle relaxants, which also direct acting. Those that are used during surgical procedures to induce skeletal muscle paralysis, this is as an adjunct to the general anesthetics. Neuromuscular blocking drugs, or NMBTs, are used in anesthesia to impair neuromuscular transmission and also provide skeletal muscle relaxation. And these drugs enable the anesthetist to perform tracheal intubation, facilitate ventilation, and to provide optimal surgical operating condition, for example, during laparotomy. And basically, the NMBD prevent voluntary or reflex-induced muscle contractions during surgical procedures. This slide actually explain in detail the therapeutic uses of skeletal muscle relaxant, starting from the adjunct to general anesthesia as an antispasmodic and also antispastic.